Hi, morning everyone. Thank you for coming and giving me this opportunity to present to you today. So my name is Kwok and I'm a PhD student with Professor David Craik at the University of Queensland. Based at the Center for Innovations in Peptide and Protein Science. So in the spirit of introducing a little bit of yourself, I grew up in Penang, Malaysia. And how I ended up in this project was several years ago. I guess my supervisors asked me if I wanted to work on the Great Barrier Reef. I took the word "on" literally and raised my hand and signed up. Little did I know that the project needed expertise in biochemistry and bioprocessing. So after numerous years now, I still haven't really been out to the reef, but I hope our work in the lab will contribute to the overarching aim of controlling COTS, or more how Dave put it yesterday, was protecting corals in general. So I won't go through the factors involved in COTS outbreak, as it's been reinforced multiple times by all the good speakers we had over the few days. The only thing I would highlight, I guess, is the COTS outbreaks are not isolated to this reef, the Great Barrier Reef, but it's a problem faced across the Indo-Pacific region in general. So current COTS outbreaks, current COTS control relies on surveys as well as divers culling COTS individually one by one. Although efficient, any methods to be able to streamline that would be greatly, I think, appreciated. So when I first started as well, I got shown this picture, and I was asked to estimate how many COTS you think is in this little coral matrix. And I can't remember the number, but the number I put out was way way lower than what it was actually is on this just one reef. So I learned from early on that COTS are cryptic and they are very difficult to find during the day, and they are mainly active at night. To compound to that problem, I guess the Great Barrier Reef covers quite a large area, and so if we can develop tools that can be complementary to the current COTS control program, I guess that would be a big help. So the project aim overall is to develop a rapidly scalable process where we could, have to be based on sustainability as well as looking at being able to fractionate large volumes of water to use as potential attractants. So I guess the first thing is, what are semiochemicals? Semiochemicals essentially are natural chemicals that are released by animals, which will then invoke or elicit a behavioral response from another animal, be it the same or different species. So in terms of COTS, we're interested in COTS-specific chemicals that we could potentially use as attractants. So I guess that's the base of, this study. So we based this off a paper that was published by UQ and Ames several years ago, where they showed that if you put a bunch of COTS in a header tank and you allow them to release that semiochemical, and if you place a native COT at the bottom, what you generally see is this native COTS would actually go and move towards that treatment arm. So proving the fact that if you put COTS together, that they release signals which will attract other COTS. But there was few complexities to these issues because the volume needed that needed to be processed is relatively large. And at a point where we were introduced to the project, some of the chromatography techniques that were used require pre-treatments. So one such example is reverse phase chromatography, where your molecules would have to be acidified prior to fractionation. But acidification itself would cause changes to the biophysical properties of molecules, and in many cases rendering those molecules to lose their function. So what we did was we selected tangential flow filtration, or TFF, as a possible solution. So TFF is a method that can be used, and it's called a soft method, where you don't have to pre-treat your samples, and you can use it, your samples as is. So just a bit of introduction about what TFF is. Essentially, TFF it fractionates based on molecular weight, so you can separate different molecules based on the size that they group in. Traditional methods of size fractionation actually will block up your column because they flow in the same direction, whereas tangential flow or cross-flow filtration moves in one direction and allows the smaller molecules to be easily, allows the smaller molecules to go through the membranes. Hence, you get separation quite easily without blockage. The other beauty about TFF is that it's very much scalable. So anything you do on a lab scale can be easily scaled up to a commercial level with minimal optimization. The other thing that we were hoping to get is something that's globally acceptable. So TFF is used widely, as shown here, biomanufacturing. So if you have your vaccine or antibody production, you use TFF right at the end for buffer exchange and formulation purposes. It's also widely used in the food and beverage industry, so you have food dye extraction or wine clarification. This is also used in waste management. So overall, TFF is widely acceptable globally, and it's easily accessible to, say, if you want to set up a plant somewhere else, and yeah, it's widely available. So how do we apply TFF, I guess, to the COTS? How do we use TFF to fractionate COTS semiochemicals? So we replicated what was done in the paper prior, which in a 40-liter tank, we placed 3 to 4 COTS, depending on the size, and in about 40 liters of water. And we did that for about 20 tanks. So what we'll do is we'll place the COTS in them, in the tanks, allow them to release that chemical, remove the animals, and there we have our COTS-conditioned seawater with the chemicals that we are interested in. We next proceed with filtration and then fractionation using TFF. And using a 10 kilodalton membrane as our first cutoff, we then get the larger than 10 and the smaller than 10 kilodalton fractions. And from there, we do another fractionation at 30 kilodalton, and that leaves us with a bunch of fractions. And in this, as shown here in this illustration, we got our molecules separated based on size. So this, we demonstrated that TFF can be used for the fractionation of large bodies of water, especially in COTS semiochemical fractionation. We next wanted to test if these fractions had any use in terms of an attractant for COTS. And using the Y-maze behavioral assay, we showed that in this case, the COTS are attracted to the larger than 30 kilodalton fraction, as shown in this graph here. Next, we looked at a few other things, such as meandering, where we show that if the COTS does not detect an attractant, it will just wander around aimlessly. But the moment you put in a fraction that contains a possible attractant, you see this drop in that meandering, and you become quite focused, and you move right towards that source point. There are other factors that we looked at as well, such as mobility, increase in mobility, and also the positioning of them, the animals, towards the treatment arm. And overall, I guess, this indicates that the larger than 30 kilodalton might contain an attractant or few of them together. We're not sure at this stage. The next thing we wanted to do is characterize what exactly is in that fraction. So we looked using proteomic analysis. We looked at what is in this fraction, and we discovered a bunch of proteins, but primarily the majority of it are proteins involved in signaling. And one candidate that sort of stood out to us is this epidiamine-related proteins that have been previously thought to be involved in signaling. As a side thing that we worked on as well is we developed a bioproduction platform that can be used to produce this protein. And for one example is the COTS epidiamine protein we have generated at large scale. So if you ever come to a conclusion that one protein is the attractant, there's a sort of a scale-up platform that's ready to go. We looked at the glycomics, and overall what the glycans tells us that this protein are highly glycosylated. Next, we looked at whether this fraction is actually toxic. It's actually toxic, but based on the brine stream toxicity, we can tell that it's not toxic at all. We then wanted to look at winter versus summer. Overall, what we found was in summer, we have an attractant in the fraction, but in winter, we don't actually see any attraction at all. So indicating that this could be winter versus summer, and summer is probably when we'll get attractants in the water itself. In conclusion, I guess we developed a rapidly scalable, easily adaptable method that can be used for fractionation. And the summer fraction is probably where we want to be looking at moving forward. So I guess I'll leave you with just a possible application. So now going back to that complex matrix, maybe we can target this to a more favorable area and get the COTS out of the matrix for easy, I guess, culling. Yeah. And I'd like to thank my supervisor, David Craik, and Cherie and Richard, who are here today, that we've been involved in the work. Yeah.
Do we have any questions? Oh. So this is a question from Slido. Are there plans to synthetically reproduce the semiochemicals?
Maybe not synthetic, but a bioproduction platform. Yeah. So definitely, if we can get or identify a particular compound. Yep.
My voice carries so worried. Do you think that that Degnan diamond is working alone? You know, that's your 30 kilodalton fraction.
Yep.
Very often these behaviors are respond to multi-chemicals. I'm just wondering if you could make your attractant even stronger if you knew what the Degnan diamond was working together with, because there's usually different
Yeah, you're probably right. We probably think it's a synergistic effect of different molecules. Could be protein, small molecules. We don't know. But with that epidiamine, we actually did express quite a lot of it, and we did test it by itself in the behavioural assay, but we didn't get any attraction, I guess.
Well, thank you for your talk. That was fantastic, York.